So again, I'd like just to go very fast over the summary of the course. Yesterday, so we had the introduction the first day, genotype networks, and then yesterday we were talking a lot about RNA folding, genotype networks, and RNA and in different models, and we stopped at population dynamics. We saw part of it, and today I'm going to be brief about what remains, because I would really like to go into the evolutionary implications of everything I've been talking about with some examples, and so I think this is fun. So let's see if we can make it to the very end. Okay, so yesterday we were talking about the dynamics, and we saw that when you have these dynamics of networks, you can have a formal description of these dynamics, and there are some final states that correspond to the mutation selection equilibrium and so on. I'm now a little bit interested in the transient. So what happens when evolution proceeds, right? It turns out that neutral networks, so again, just to fix ideas, I had three levels, or I separated evolution in three different sets because the structure of networks changes slightly. One was evolution on neutral networks, where all genotypes map to the same phenotype, so they have equal fitness values in a given environment. Second set, this is when you have adaptive evolution, and then you jump from phenotype to phenotype. And a third group of say adaptation when you have environmental changes. So in the first case, when you have these dynamics on neutral networks, it turns out that because neutral networks have a community structure, so you have regions of these networks which are more tightly connected within themselves than to other regions of this network, then you have this community structure, these blobs, these groups that are usually related in RNA to composition of the sequence. And what happens is that you have discrete adaptive jumps if these communities are really connected, which is the case. And this case is much more frequent. The longer the sequence is, the larger the network is, right? It's more easy to have a hierarchical structure in those networks if they are very big. So even if you are keeping the same function in your network, just for instance, recall that microRNA that we saw, that having the same structure, probably the same function, can have adaptive jumps towards higher values of robustness, so an improved neighborhood where mutations matter less, right? So, and this progression, due to this community structure, occurs in jumps, in discrete jumps. So you have this sort of punctuated equilibrium even in neutral networks due to this community structure. When you go to adaptive dynamics, meaning you have to change phenotype, so you can imagine again the same dynamics that we were discussing. You have a population of N molecules that you choose at random or in any way you want. So this is your initial conditions. And then you let the populations evolve towards a prefixed goal structure. This means this is a practical way of implementing the fact that you may be sitting in a secondary structure, which is this violet phenotype, and then you want to go to the red phenotype because this is the one that you know is fittest in the environment you're working in. So this is a practical way of saying, okay, how I go from here to here. So I will accept mutations that take me to phenotypes which are a bit more close to the goal phenotype, right? Again, I will measure distances between secondary structures, and I can implement that in my evolution by selecting depending on the distance to the goal phenotype, right? So you let this population evolve then, and you will have, in addition to this drift on the neutral network, which corresponds to the previous picture, you will also have jumps to phenotypes of higher fitness value, right? So this is what I was explaining, was what is written here. And if you have a suitable mutation rate, of course, it's you don't lose the structure of your population, and you allow that mutations are fixed somehow in the phenotype. What you have, so this would be different secondary structures. You can calculate here the distance. What you eventually have is a convergence towards this goal state, right? This could be selective function. So the selective pressure, you could implement it as an exponential with a given parameter beta of the distance to the goal structure. If you do like that, then you have this kind of evolution, where your population spends sometimes in the neutral network of a given phenotype, but then jumps to an improved phenotype closer to the goal, also spends some other time in another phenotype until it eventually, through these successive jumps that now correspond to phenotypic jumps, to changes of phenotype, eventually reaches this goal structure. And here, if you maintain the evolution for long enough time, you would have this evolution towards increased robustness that we saw in the first case when we were dealing with neutral networks, right? So also in adaptive evolution, when you change phenotype, you do have this punctuated dynamics. So this is intrinsic to the structure of genotype spaces, to the way in which they are fragmented. There is also something interesting that happens concomitantly. So at the same time that you have these searches in neutral network and jumps to different phenotypes, which is a systematic growth in heterogeneity that typically occurs once the jump has occurred. So you have this you find this new phenotype, for instance, here in this jump. This phenotype is better, so closer to the goal phenotype. And then you have a bottleneck in genetic diversity. This is a, say, schematic representation of genetic diversity of my population. And then I have a decrease in this diversity because I typically enter the network through one of few nodes that are in contact with the network of my previous phenotype, right? After that, I have an expansion in this neutral network, and I have so my population drifting, expanding, and at the same time, in an, say, unsupervised way and non-directed way, searching for different phenotypes or getting in contact to different phenotypes, right? In the way that we were also visualizing yesterday with those networks in contact one to another. Again, you find that this new phenotype, which is better, you have again your genetic bottleneck, and then you have again the expansion. So you have this series of reductions of diversity, bottlenecks, expansions in diversity, selection of a new phenotype, and so on. Yes, Pratyush.
So I just wanted to ask, how exactly are we defining this heterogeneity or diversity here? Is it the number of neighbors, neutral neighbors, or how exactly are we defining this?
No, it's genetic diversity. So this could be pair distance between all the sequences in your population. So if you take all your sequences and you calculate how far are one from another, right? In positions, in genomic positions, in nucleotides, and then you plot it here. So if they are very close together, this would be close to 0. I mean, the scale is not what is written here, which is for this plot, right? This is just an illustration that you have some separation in the genetic similarity between sequences as evolution proceeds. So one way to quantify that would be the pair distance, the mutual distance, Hamming distance between sequences with taking all pairs in the population. And then you see that it diverges. I'm going to show another plot where this can be more explicitly seen in a moment, okay?
Right, so we are starting with a population of sequences in the first place, is that? Yeah. Is that the case? Okay.
Yes, yeah, like here, you have a population of N sequences of RNA.
Right, okay, yeah, right. Thank you.
Right, and you choose them the way you want, because actually, from the very beginning, since you are selecting for a goal structure, just immediately, if you have a high diversity of structures, but they are very far from the goal, you already have these genetic bottlenecks, and you just select those that are closer to the goal. Yeah. Amir?
Okay, thank you.
So I was just saying, so this is with respect to one gene, right? One structure. What if there were more structures? Would you still see punctuated evolution?
Yeah, then you may see something very funny, which is an attempt to optimize both structures. I haven't done the simulations with 2 structures at once, selecting for both. I don't know. It can be either that, and this can be a question for you if you're interested in computational evolution. It could be that the population segregates into groups that go to one or another structure, or it could be that the population makes an effort to sit at regions where the 2 phenotypes are in contact. This is what happens, for instance, with molecular switches. Actually, there you are sharing two structures or two functions, which are very close. And either by changing the environment or sometimes by changing just one or two positions in your sequences, you go from one structure to the other. So I don't know which would be the solution to this problem. What I know is that when you present the system, the population of sequences with the two sequences alternating, so first one, then another one at a given time that allows some minimum optimization or evolution towards the structure, they tend to go to these regions where the two networks intersect each other, right? So that's a good question. And yeah. Okay. So another important point is what happens, and here I want to go back to this idea of so phenotypes that have peak networks, phenotypes that are easily found. For instance, this is a classification of different structures of RNA secondary structures. So those ones are quite common. Well, this is I don't know where is this. This might be the excuse me, the open structure, which is down there. Okay, then you have hairpins. Hairpins for short sequences are very easy. Hairpins with two hairpins, right? Two hairpins, three hairpins, four hairpins for 4 loops. Yeah, double. So sequences with two stacks, with one stack, two stacks, three stacks, and so on. So this is structurally the complexity here grows somehow, and this also means that growing structural complexity diminishes the size of the network of genotypes of each of the sequences, and also the probability of finding those structures, essentially, right? So what you see here is for different values of the mutation rate, how many generations it takes to fix one of these secondary structures. So in the previous plot, imagine I fix it here, this secondary structure, doesn't matter specifically which one, but of course I have a number of intermediate steps, and it takes a time that I can quantify to reach this goal structure. So this is what I'm doing in the next plot, and I'm counting the number of generations. This is arbitrary units. In the next plot is generations that it takes to fix structures of increasing complexity, more or less. So for very complex structures, you see that it takes very long time, and also the region of mutation rates in which you can fix them is very limited. This has to do with the idea that I discussed yesterday, that given the mutation rate of an organism, say here, there might be phenotypes that are lost under evolution, and this would be the case of this not so abundant phenotype. You have a too high mutation rate for the organism, and then you can only fix phenotypes which are above a threshold of abundance, say like this hairpin 2, right? And the others, right? This is very rare, this is not fixed, and so on. And you see how easy it is to find phenotypes that are simple, in this case hairpins, right? This is, I think, for sequences of length 35. This is one of the examples I have also shown. So they allow certain structural complexity, but it's very easy to have simple hairpins in just a few generations, in 2 generations, just for free. This is a sort of function for free. You just 4 random sequences, and there are some hairpins there. Other structures are more complex to find. This means that fixing a secondary structure also determines the mutation rates that one can work with, and has an impact on the evolutionary time to fix those phenotypes, right? When we do this kind of evolution, we also see many instances of convergent evolution, because when you start your population from any place in the space of sequences, where in the goal network it eventually will find, so the pathway to enter this goal phenotype can be different in any of the simulations that we perform. So if we replay the tape of life and we start with a different initial condition, the pathway I will follow will be different. And although eventually I will get the same phenotype, so I have this convergent evolution, the composition of my population can be very different, right? So this is an illustration of this kind of behavior. What I'm plotting here, and all 4 plots are equal for different types, for different mutation rates above and below, and for different secondary structures, right and left, as I show here. Every histogram, what it shows is the Hamming distances between sequences within the same population, black histograms, right, at 2 different times of evolution. At the initial evolution, this is when the population has experienced the genetic bottleneck, and thus is concentrated and is not that diverse, genetically speaking. And later in evolution, when it has already spread on the neural network, and you see that these histograms are broader, right, have a larger sigma. This is another representation of this qualitative picture that I've shown before, that someone has asked about how to quantify it, right? Here I'm more concentrated and I'm more spread. But this is within a population. What happens if I repeat my simulations 1 or 2 or 3 times, and I calculate the distances between these populations? Then genetically speaking, these populations differ one from another, although they coincide in their phenotypes. I have 2 questions, so please. I don't see the names of who is asking.
Okay, so I have a question from the previous slide, if
Yes,
that's okay for you. So in the previous slide, yes, so I was expecting a monotonically decreasing curve, because as the mutation rate increases, it might become easier to reach to a particular network, particular type of network, which seems more or less the case for less complex phenotypes at the bottom. But however, it's kind of non-monotonic as you go to this more complex structure at the top. And I don't understand, like, what's roughly the reason? Is it selection or what? I mean, I don't know.
it's a combination of finding the phenotype. Since you are doing random moves in your space of sequences, then there is, if it's not abundant, it will take time to find the first sequence that belongs to the genotype network of that phenotype. That's the first effect. The second effect is if once you have found the phenotype, you can fix it. So this is a sort of quasi-species transition, right? That occurs at different rates of mu depending on the phenotype. So if you find a sequence that is in the genotype network of your goal phenotype, but your mutation rate is so high that prevents it to be transmitted to the other sequences and to be fixed, essentially you're not there. So here you have in all instances a substantial fraction of population in the goal phenotype. If this is not the case, that I'm not plotting it here. Here, for instance, you have gone through the error threshold. You cannot fix it, and this error threshold depends on the phenotype. Okay,
so these are the generations for like to be fixed, not just to reach
not just to find one, but also to get it fixed in the population. It's find and fix, right? Sorry, perhaps I was not clear about that. Thank you for the question. Yes.
Thanks.
And second question? No? Okay, then I continue. Just raise your hand again if you have any other doubt. Okay, so I was saying here that so when you work, so the conclusion of these simulations, and also an idea when we look at different populations at the molecular level, is that when you go look within a population, this would be the black histograms, then you have some memory, so phylogenetic memory, because sequences come one from another, right? And then you have a relationship among those sequences, and they are close in sequence, and you can relate them by a replication, by a descent relationship. But when you go to different populations at the molecular level, perhaps it's nonsense to try to force phylogenetic similarity, because you can have convergence in the phenotype if those phenotypes are small, if those sequences are relatively short, right? And you don't necessarily need to have a common phylogenetic origin. So although this, of course, is true for high organism and for very complex functions, and we have seen repeatedly in genes, when you go to the molecular level, when you think of microRNAs, when you see that the hairpins are sort of costless structures that are continuously produced through random RNA sequences, why do you need to look for phylogeny there? So these functions might emerge once and again along evolution if the selective pressure is there. Right? I will go again over this argument when in the third part, when we see examples in nature. Okay, the third case of evolution of dynamics on networks that I wanted to discuss was the case of adaptation in a changing environment. This is a very simple computational experiment where I have devised it. I have designed a network. In this case, is a scale-free network, and I have let my population evolve when the network is neutral. Right, so as expected, it goes to the highly connected regions, and this is the final distribution of abundances that I have in my network. To fix ideas, this could be the mutation selection equilibrium. My population is heterogeneous at equilibrium, and this corresponds to the eigenvector of the transition matrix, of the adjacency matrix in this case that we were talking about yesterday. Now, in these two pictures, what I have done is for each of the nodes, which is a sequence with the same secondary structure but different folding energy, I have also included the folding energy for each of the nodes, and I have selected over the folded energy. Just in purpose, I have situated sequences of low energy here at the top of the network, and I have changed the relative value that selecting for high robustness has in front of selecting for low energy. So I have a parameter that tunes the relevance of these two phenotypic quantities. And what we see is that the population, of course, that essentially only selects for robustness if I don't pay attention to energy, when all my attention is focused on energy, then experiences a transition from sitting in this region of high connectedness to this region of low energy, right? Okay, so there are important quantities here that tell me that there might be a sort of critical transition between these two environments. The first one is how dispersed the population is. This is the first plot here. This is a parameter that I'm tuning, which is, well, beta is the parameter that gives me the relative weight of one environment in front of the other, and mu is the mutation rate. And we know that when we use the relationship between the two parameters, we have more or less the curves collapsing on one same curve, right? So this is the dispersion of the population. This was the first case. You see that I'm increasing this beta parameter. It moves, although it keeps more or less the same value. And then quite suddenly, it goes to high values and returns and moves the population to the other region of sequences, right? So this transition is really fast. You see, so it's not a smooth transition although I'm moving very slowly. So step by step, the parameter that selects one situation in front of the other. This is also reflected in the time it takes to reach equilibrium. I have used here linear scale. If you use logarithmic scale, you see almost an exponential increase. A power law increase near to the transition. And you see this divergence in the time it takes to reach equilibrium. So when you are close to the transition, the population is sitting in highly metastable states that could fall on one or another side, but essentially it takes really long to achieve equilibrium. So again, here you have a sort of punctuated dynamics where you go from one state to another in a very short interval. Might be time or change in the parameters in this case. Right.
Yeah,
Let me.
Could you please explain this beta parameter again and these transitions? Again, I'm sorry, I missed this.
I haven't shown you the equations, but this is something similar to what I was showing here, right? But instead of having just the distance to the goal structure, what I have is a compromise between the two variables in my system. So beta 0 tells me that I'm selecting only for one of the two parameters, and beta 1 I'm fully selecting for the other, right? And depending on how close my phenotype now has two terms, right? One for each phenotypic trait, one for robustness, and the other for energy, right? And so this is the idea I think you can, here's the paper where we did that, right? And the idea is you have a way to balance between both environments by going from 0, only robustness matters, to 1, only low energy matters, right? So, I mean, actually, in practice, any function that you can devise where that has these 2 limit behaviors is okay. And in particular, we have chosen functions that interpolate smoothly between the 2. I think I'm going to give another example of that. Okay, so since this was a synthetic example, we also worked with something which is quite well known, which are Kauffman's NK model. That actually what it does is you have a network of sequences, and then it assigns a fitness value in a certain correlated way to the neighbor. So it's not random values, but it's nice because you can set how correlated is your landscape. So it's a rough landscape with short range correlations. And we have studied also evolution, same that I did in the previous slide. We have studied evolution on such networks with these fitness landscapes. And it's really interesting what happens, which has allowed us to see how are those fluctuations and those changes in the composition of my populations, the movements across these landscapes. Here we had the little amount of noise, and what we were monitoring is between 2 different landscapes, meaning 2 different instances of this NK model, right? And interpolating the value, the fitness value of each of the nodes in my network with a little amount of noise. What we were monitoring was changes in fitness. This was calculated as the projection of one vector over another. So your landscape can be one vector, and your initial landscape and your final landscape is another vector. And for instance, you can calculate the cosinus between the 2 vectors, and it gives you a measure of similarity. So they start being identical, and then they part from each other. Here you see this noisy effect, right? But this occurs in a very smooth way, right? And here you have the difference in composition of the population in black, again, the cosinus distance of the stationary state of the mutation selection equilibrium of my population along the environmental changes that I represent here. So you see there is little environmental change, and you can have sudden responses in the population that moves so to different regions of the sequence space. So you have these shifts in sequence composition of the population that can be very rapid. This is a distribution of the histogram of the changes in a time t with respect to time t+1, right? You see that for the case of the environment, this is a very narrow distribution, a Gaussian distribution, because essentially you just have here noise, the effect of noise. And here you have the distribution in changes in the population, which is much broader, but especially here in the large changes, right? You see this close to power law decay or fat tail decay in the relative change of the composition of the population. So you have a smooth changes, but you have sudden responses of the population. This is another way of seeing the same phenomenon. And again, as I've been repeating all through the course, so the idea is that this phenomenology is not very much dependent on the model you use. I can see it with RNA, and I can see it with Kauffman's model, and I can see it with artificial networks, as I was doing in this experiment, in this computational experiment, right? So the phenomenology is the same. Right? So this should be a robust property of my systems. Interestingly, since in the case of Kauffman networks, I have here a Kauffman model, NK model, I have here some noise. I can calculate a number of early warning signals. So changes in my population that are alerting me of the near presence of a sudden change in composition. This is, for instance, the distance between one population in 2 successive states, right? And what you see here is the difference is really large. So at the beginning, I don't see major changes because still I am sitting far from the transition. But as I approach the transition, I see that in subsequent time steps, I can have very sudden changes until eventually I go through the transition and I sit in the new stationary state. Right. So this happens when the two eigenvalues of my transition matrix are very close one to another. Right. So the largest eigenvalue becomes very close to the second largest eigenvalue. And if you go back to what we were discussing about the time to equilibrium, you will see that in the denominator you have the difference between the two largest eigenvalues. So here is where you have divergence in the time to equilibrium and all these complex and very nice, I think, phenomenology for these transitions. So eventually, yeah.
Yeah, sorry. So I thought the transition matrix was stochastic. So, but seems like the largest eigenvalue is not 1 in the second figure.
Yep.
So it wasn't stochastic, the transition matrix?
You have an addition of noise, but the main effect. So you need noise if you want to see this kind of effects, because otherwise it's deterministic, the transition. You don't have early warning signals or something similar. You need some noise for that, right? But the transition matrix is deterministic. You add the noise on top of that. So still you have a dominating effect, which is that of the M matrix, which gives you the main value of the eigenvalue, right? And when you add the noise, you have some departures. But since you are changing from one landscape to the other, right? When this happens, you have more or less a linear change in the dominant linear change in the eigenvalues. Yeah.
Thanks.
Sure. Okay, so and finally, again, if you have here a time to equilibrium that is diverging, and we believe it is diverging, although with finite systems you can never be sure, right? So it's increasing so much. It turns out that it doesn't matter how slowly your environment changes, there will be always a time at which the environmental variation is shorter than the time you need to reach equilibrium. Is that clear? So here you reach equilibrium fast. So if your environment changes slowly, there is enough time for adaptation and to reach mutation selection equilibrium. But here it is no longer the case. So my population can never get to equilibrium before the next environmental change arrives. Therefore, if my environment changes at a given speed and eventually my population cannot get adapted to it, and I have this hysteresis where my population is delayed in adaptation with respect to environmental changes, and eventually, of course, it will go to the other state and the other way around. If I start from this state, I cannot keep pace with environmental changes. I'm delayed, and I'm eventually going to the other state when adaptation times are short enough. This would be the transition for infinitely long times, right? This black line here, but for any finite time, I will have hysteresis in my population. This also has a side effect, which is the possibility of extinctions if environmental variations are too fast. So my population can get stuck here, and if it's not able to jump to the other state because of finite size effects in my population, I can get extinctions. Right? We studied this also in a paper that we published later on the topic. And I'm not going to discuss about it today. So this is another representation of how these smooth changes in the landscape lead to sudden responses in my population. Another way of visualizing the same here in the top plot and CEG, what you have is the composition of your population, the abundance of the different nodes. They have been ordered in an arbitrary order for a given simulation. And then you see that this node was the most populated node at a certain point, but all of a sudden it jumps to another region of the network, and this is close to the transition where you have this metastable state that you try to be in both places at the same time, right? With an increase in the dispersion of the population, in the heterogeneity of the population. But if you look at the fitness landscape, you don't see that large variations. So when we interpolate, you still have this node, which is the node of highest fitness, meaning highest reproductive value in this case, right? But still, the population is not mostly concentrated here because of topology, because other quantities matter, and the connectivity and the nature of the neighborhood of other nodes makes the population jump from this node, which has the highest fitness, to this other node where it doesn't replicate so fast, but the neighborhood is more favorable to maintain a high density of populations here, right? So you don't see major changes, but you do see them in the population. So to sum up this part of the of dynamics, what we have is different kinds of transitions that actually all of them are punctuated. In neutral networks, this is a sort of entropic transition when you move to regions of lower connectivity to regions of higher connectivity, right? In adaptive transitions, you go across a fitness barrier, which is positive, right? So there is a trend towards reaching phenotypes with higher fitness if they are also sufficiently abundant, right? This has to be kept in mind. And when you have complex fitness landscapes where so fitness is distributed in as it is, right? Then you have a complex interplay of both quantities. So in the first case, we would have only that the adjacency matrix C in our notation of yesterday is relevant. In the second case, we have a competition between networks with two different adjacency matrices and different levels of fitness. So it's essentially a two-block matrix with different connectivities and different diagonal values, which would be the replicative ability of the nodes in each of them. And there is a whole theory that we developed, well, colleagues of me developed to visualize or to understand these kind of transitions are competition between networks, right? And in the third case, then you have to consider your adjacency matrix, but also your fitness matrix as well, because you have all different values in the diagonal, and then you cannot disentangle one from the other, right? Both at the same time, topology and replicative ability matter simultaneously to tell you what's the final state of the population. Okay, so with this, I finish the dynamical part. If there are any more questions, otherwise we will go to the examples. No? Okay, then I will give you a number of empirical examples of how we see all this phenomenology I've been describing in the molecular world. First, we talked at some point about the proximity of phenotypes in genotype spaces, right? There was the idea that in these spaces of high dimensionality, two phenotypes can be quite close one to another because networks are very large and networks percolate the space of genotypes. Okay, so is this something realistic? So what happens when my phenotype is a real molecular function? So there was a very nice experiment carried out by Schultes and Bartel in the year 2000, where they considered two RNA folds. These are 3D folds, not 2D folds, because they are well, because the 3D structure is involved in the functionality. So the first molecule was a class 3 self-ligating ribozyme, which was evolved in vitro, right? So no natural analogs. The second molecule was a part of hepatitis delta virus, which is a self-cleaving ribozyme, and is natural, occurs in this virus, right? And both have the same length. Just to be sure that the similarities, there were no spurious similarities, they made sure that in this selection, no more than the 25% similarity expected by chance was found. Of course, they didn't have any shared evolutionary history, and there were no fortuitous structural similarities. So they are quite different, these two molecules, right? So the question they posed themselves was, is it possible to reach, to find where the neutral networks of these two molecules, of these two functional molecules intersect? So where are they so close that with one or two changes in positions, I could jump from one functional molecule of this kind to one functional molecule of the other kind? And this is what they did. So they managed to measure the performance of each of the molecules and to incorporate mutations that progressively were making the two sequences to be closer and closer and closer until they reached this intersection point. So at the very intersection point, so it was not possible with the same sequence to have the two functions, but they were two mutations away and still performing relatively well, right? They changed it over 40 positions in each of the sequences of length 89. So you can imagine it was about half of the sequence was changed, but still the function was maintained, and they eventually got these two molecules where, with the exception of these two mutations, signaled here, this U is a C in the other molecule, and this C is an A in the other molecule. You see the other way around here, right? With these two exceptions, they could get the ligase fault or the HDB fault, and they were both functional. And you see the tremendous rearrangement in structure that changes from one molecule to the other. This is here illustrated in colors. So you see the stacks of the same color here. For instance, this one is divided into a small part here, another part here. With the blue stack, and the other is sharing two stacks that were previously at different parts. So this was, in my view, one of the nicest experiments I've seen. There was some continuations later, but this is a proof of concept, a wonderful proof of concept in my view, of the existence of these very large neutral networks and the possibility of really jumping from one phenotype to another with just one or two mutations, right? So phenotypes are very close somewhere in sequence spaces, at least for RNA. Okay, the second example that I wanted to share was the case of punctuated adaptation in influenza. So the evolution of influenza occurs on a complex landscape, of course, with many different fitness values. And from season to season, there is also a situation where the environment changes because the immune state of the hosts of influenza changes, right? So this is an environmental landscape. And also the sequences change because they are incorporating mutations steadily, neutral and non-neutral. And right, we expect then this kind of probably evolution of adaptive evolution through seasons. And this is what occurs actually. So this is a plot just to illustrate the speed at which influenza incorporates mutations, and also colors indicate antigenic changes. So essentially within each color, it's incorporating mutations, but it's not changing the phenotype. So if your immune system was resistant to this strain of influenza, it's also resistant to this strain here in the same color, but will not be that resistant to the next mutation because there has been an immunogenic change, right? So this is the structure of influenza virus, and you can have mutations in different positions where so that make your immune system not able to recognize the virus, right? Especially in the two proteins of the capsid, neuraminidase and hemagglutinin, which stand also H and N for the typical, so for the names of many influenza strains. H3N2 means specific mutations in hemagglutinin and neuraminidase. Okay. So, well, this is also an interesting virus in itself because it has 8 chromosomes and can undergo also this shuffling of chromosomes within individuals. And this is one of the problems also with recombination, because you don't need actual recombination, but you can just shuffle the chromosomes and increase diversity in a very rapid way. But that's another point. Okay, so some people from the group of Mercedes Pascual in the States studied the evolution of the diversity in influenza for many seasons, for many years, and also studied the antigenic properties of the virus. And they found that there was this drifting and switching that we were describing also in RNA populations. So what you see essentially is that populations expand, influenza populations expand on neutral networks, and now the phenotype is the antigenic type, right? So the recognition of the immune system. So that's the phenotype here. And when the selective pressure is such that not enough susceptible hosts are available from one season to the next, something that happens, then it has to jump to another antigenic type. And when this occurs, you have a genetic bottleneck, and you have again an expansion on the new neutron network of the new antigenic type. So you have this stasis and these punctuations, these sudden transitions when a new antigenic phenotype is found, right? Okay, now I have a bit longer story about how relevant all these ideas might be in the RNA world. And for that, I'd like to give a brief introduction to the RNA world by starting with a brief chronology of life. Okay, so this is the story, the history of our Earth. It starts about 4.5 billion years ago until present. And there are a series of important events along this history, right? Formation of Earth and Moon at the very beginning. Our Earth was very hot at that time, but relatively soon after, at 3.9 billion years, you already have the oldest sedimentary rocks, meaning that the meteoritic bombardment, which was keeping the Earth warm and also had a very high volcanic activity, was already stopping. And it's surprising that about just 0.1 billion years later, you already had autotrophic carbon detected in these rocks. There are still no evidences, direct evidences of life, but an accumulation of the isotope of the carbon that living organisms use, right? Eventually, we have a detection of the oldest microfossils about 3.5 billion years ago. There are some other series of events, like oxygenic photosynthesis, the first eukaryotic cell, and multicellular organisms much later to give rise to animals about 700 million years ago. Okay, so somewhere between the time when the Earth was cold enough and the appearance of the oldest microfossils, there should be the origin of life, right? I'm assuming that life appeared on Earth, so no theories about panspermia, which just puts the problem somewhere else and doesn't solve it. So let's assume that life appeared in this, well, short or not, you know, I mean, the time scales depend on the processes that we know that can generate complex chemistry and complex life. If we don't have such a process, for me, it's very difficult to say whether this was fast or slow, right? We don't know any single pathway that can go from inorganic to organic. So I don't dare to qualify this as fast or slow, but anyway, somewhere here, there was the origin of life. So if I expand this region, our idea of how events could have occurred was starting with a prebiotic chemistry, which was could have been really complex because there were no organisms taking the energy of such molecules or degrading any complex product of chemistry. And so exploration was proceeding in many different environments at once, right? Probably there was later a sort of pre-RNA world with polymers and some more complex chemical behavior. Then to go to the RNA world, this is one of the possible sequences before we get to work with the division of function of DNA as coding for proteins, and proteins performing the chemical functions, right? And then once you trigger self-replication, somehow the Darwinian evolution is unavoidable. So you would steadily go through, not that easily, but we have a better picture of which could have been the steps that led to complex life and to its diversification. So people very often use this bottom-up approach, and I'll talk a little bit about what happens here. And some other people are focused in this top-down approach, and they look for minimal cells and so on. But there is a huge gap still between what you can get by starting with inorganic material and what you can get if you start with fully functional cells, right? So there are many steps in between that we are not sure about what happened. So for the bottom-up approach, you have certainly heard about Miller's experiment and the production of many amino acids and other monomers in a quite simple environment. So it's for me, it's not that difficult to imagine that in a real setting with the size of Earth, a lab the size of Earth with so many different environments, so many complex, much complex chemistry was going on before life appeared, right? So there is anyway this long way to life when you start with these organic monomers, which is called the primordial soup, and some precursors, and then you go to Luca, and in between many of these things happen. And well, just a little joke here. So because then a miracle occurs, something happens, and you go to cellular life. There should be many things in between, right? And some of these things in between have breached the appearance of cells. And in particular, we believe that RNA world, and so complex chemistry on surfaces and metabolism in simple vesicles. I mean, there have been significant advances in understanding some possible processes to increase inorganic chemistry towards self-replicating vesicles, say, in the last 10 to 20 years, right? Still, there is this problem, right, of how if you need DNA to make proteins and proteins to make DNA, how do you solve this catch-22 problem? And the agreement is precisely that it was RNA performing both functions. RNA can code for so can be a genetic code, can be used as a template to transmit genetic information, and can also perform the chemical functions itself, right? And we still see many instances in extant life where RNA plays these two roles, and so it has not lost this role, and it's quite easy to accept that this it could have been the case that only RNA or PNAs or similar easy to produce polymers were performing these both functions in this RNA world, right? Still, the question remains: how can long informative polymers emerge and persist in the absence of faithful template replication? Because another problem here in between is that you should have, so when you think of how the genome replicates, you need a complex machinery to do it. So more complex even than the sequences it sometimes replicates, right? So how can we get there if we do not yet have template replication? Okay, the first part, which is the polymerization, to polymerize this, to obtain these long polymers. This is relatively simple if you do it in mineral surfaces and with, say, thermal cycling, or in this case was wetting and de-wetting cycles. You can get long stretches of RNA up to 100 mers, which is not bad. So you can have longer molecules, but of course these molecules are random, or it's in sometimes the composition is skewed, because these surfaces or the what catalyzes the formation of these covalent links so prefers one type of nucleotide in front of another. Okay, that's fine. So we have random or quasi-random sequences. What can we do with these sequences? As I was already saying before, it's very simple to get for free hairpin-like structures. When you fold short RNA sequences, very often you get hairpins. So if they are very short all the time, so all the sequences that fold are hairpins, and as you increase the length, you can have progressively more complex structures, but still these rod-like structures, hairpin with a loop at one of the extremes, these are extremely common structures, right? What's the, well, let me just be a bit more quantitative about that. So this is again the case of sequences of length 35, and if you fold them, for the most abundant structures, they are all hairpins of this kind, right? A stack finished by a loop with some dangling ends of different type, but this is what you have, right? The second most abundant class of structures is formed by 2 stacks with a loop in the middle. And the third most abundant class of structures, and you see that it is fast because it starts already in ranks about 10,000. So the 10,000 most abundant molecules are either hairpins, simple hairpins, or hairpins with a loop inside, right? Is this these triangles here? So this is the third class, a double loop, the double stacks. Okay, so if you're producing monomers of length, say 50 in average, what you are going to get is an incredible amount of hairpins when they fold under simple thermodynamical rules. I don't have any kind of evolution of selection yet, right? Just random polymerization and folding according to thermodynamic. This is a bit more so quantitative about the relative abundance of different kinds. You see for the length 35 that I was mentioning, there are very few sequences that remain unfolded. In contrast to the case of length 12, this is 15, but this is very close. And I told you for length 12, we had up to 85% of the sequences not folding. Here we have 66. And when you go through a certain threshold, you already have a few sequences not folding. You have mostly these stem loops and hairpins, right? This similar class, and double stem loops with a lower proportion, right? And of course, the relative fractions change as you go to longer sequences, but if you're around those values, then you see that you get lots of hairpins. This will become important in a moment, because it turns out that once you have this random polymerization and you have this variety of secondary structures, it cannot be discarded that you have some selective pressures that simply occur by condensation on surfaces. Some of them can attach to surfaces, some of them cannot. So you have a sort of passive selection. But what hairpins do, and that's the very important point, is that they catalyze ligation reactions. So there are many examples of hairpins doing that. And if you have a random pool and you have your hairpins, there will be a preference to ligate reactions, to cause these ligation reactions, and therefore to generate longer molecules, right, that can fold, perhaps maintaining the structure they had in the partial structures they had previously. Okay, this is just a possible scenario, but the most important point here is that you can ligate these molecules, and therefore functions that may have been passively selected in one environment can be united, linked, joined to functions that have been passively selected in another environment. Right? And also, it's very important dynamical property of this process, which is that this is a sub-exponential process. Since you don't have replication, you don't have exponential growth of different types, and you don't have suppression of one type in front of another. Since you don't have this suppression, you have coexistence. So the diversity of molecules that you can have, of secondary structures in particular, in such a scenario without template replication still, is much higher and allows you perhaps to explore a larger fraction of the space of function that it would be if you have already exponential growth or template replication, which is the same, right? So in such a scenarios, there could have been other processes of selection and compartmentalization. We know that with so with this kind of acids, of lipids, you can form these bilayer vesicles that are just assemble spontaneously and are hydrophobic in the inside and are hydrophilic in the outside. With these compounds, you can form micelles and mineral surfaces, and different environments can also promote the generation of chemical diversity and also encapsulation, right? So to form these compartments where you could have different processes at the small scale going on inside, and then I mean, many people also have talked about the selection of these possible compartments in a passive way, so without still replication, which could have been important so mechanisms at these early times of life. So we had also the idea that a process of this kind could have led to the emergence of the first replicators by randomly polymerizing your monomers that come from inorganic chemistry, either on surfaces or inside vesicles. You could have of course, you would have this folding of oligomers of polymers, and then you could have these ligation reactions due to the free presence of hairpins, therefore longer molecules that attach to each other, and some passive selection of certain properties. And when you do this massively and at once, it cannot be discarded that can lead towards a molecule that can first polymerase these so the oligomers or the monomers that were in the environment, and also to develop some preference to do it in a template fashion, template-like. So this would be the real so qualitative change in evolution from inorganic disordered somehow chemistry to something that can start replication. And this is actually the real breakthrough. Once you start self-replication, I believe that then any little advantage that you incorporate in your molecules, in your populations, will be fixed through natural selection, which starts to act at that very moment. And so it's not that complex to get a complex function. Yeah, it seems to be relatively simple. Okay, I think I will skip this part because I will not have time otherwise to talk about some other topics, which is essentially a realization of what I was saying about selecting different phenotypes, different secondary structures in different environments, trying to ligate them so these two environments combine to each other. And then the question is, how likely is it that I maintain the functions that I have independently selected in a new environment? So we did some simulations with that, and we saw that it was, despite the fact that mutations might affect largely RNA, still this modular evolution is something very so that would maintain the properties of the individual molecules and could have been important in the generation of more complex molecules, right? Once you have template replication, this modular evolution also has some advantages, which are that the time that you need to attain a prefixed secondary structure decreases because you are only looking for small motifs, which are much faster to find than large ones. You have a larger fraction of sequences folding into the correct structure if you use modular evolution instead of using direct evolution to the complex, to the largest secondary structure. And also, you somehow avoid the problem of the error threshold, because you can tolerate higher mutation rates at a moment where correction mechanisms were not yet in place. So the length of molecules that could be replicated was relatively short, right? But if you do short plus short plus short and combine the products of these independent evolutions, then you can get into something complex, right? Without the need of directly developing this fully complex and long molecule. Okay, so this was one of the points. And here, regarding what we have been discussing, so this fact that you get the hairpins for free, because this is the most abundant phenotype, and they are produced, even the same hairpin is produced from many different instances of sequences. So their phenotypes are very large. This is say something that is there for free that could help it in the initial evolution of life in the context of an RNA world. Okay. I think this is the last issue I wanted to discuss, which is how do we empirically reconstruct genotype networks? Because we have done it computationally, but when we go to nature, the sizes of populations that we have are much below, well below the sizes of networks. I mean, you remember the names, and it's not something that we can achieve. But still, there are some instances in which we can get a sufficiently large number of different sequences of populations of viruses, and we can try to see where those populations sit in my space of genotypes and how they move. For that, in association with some colleagues, we have studied the Q beta phage, which is a simple virus that infects bacteria, E. coli, in the lab, and has 4 proteins. This they are shown here, and 2 of the proteins overlap. So one stops before, and so the other goes a bit longer. You have here a non-coding region that is also useful for to complete the viral cycle. And yeah, so this is a relatively small virus that you can adapt relatively fast. So what we did was, so the first question was, can we see any of those dynamical transitions that we have been describing in theory in a virus, which is the faster evolving organism that we can use in the lab, right? Can we see these transitions? Could we see this punctuated equilibrium at the molecular level? So first, there were a series of adaptation experiments where they started with an ancestral population of viruses, and they passaged it through time, and they attempted to adapt the virus to different temperatures. So the environment, the different environments were different temperatures for the virus. This is a summary of the experiments that were performed, and the temperature I will only briefly discuss. So there are actually at present there are still more of them, but so the virus is optimized. Well, optimized. It's adapted at 37 degrees in the lab, and so with this ancestral population, it was forced to evolve at 43 degrees for 60 passages. 60 times it was passaged from flask to flask with a number of replication cycles in each passage, to 40 degrees, maintain it at 37 degrees. You see, sometimes you may believe this doesn't make sense, but now you know that there should be some expansion in the neutral network of the virus, right? When you do this experiment. Or to lower temperatures, 33 and 30 degrees, right? There are other set of experiments, but essentially what we have is that we take the virus, change the temperature suddenly, and just let it evolve in the new environment. Our initial interest was to see what was happening along evolution. Was the adaptation of the virus with a bottleneck, punctuated? When did it change? What kind of mutations were fixed? What was the final state? Was this final state close or far from mutation selection? And the same at other temperatures. And also we wanted to compare across environments. So if we would be changing the environment here, what kind of differences would we see in the composition of my populations in these final states? Okay, for that, what we did was to select 3 regions in the genotype of the virus. This marked here R1, R2, and R3 of about 200 to 300 nucleotides in length, and with deep sequenced populations. This means that we made a single molecule sequencing, and we got a large number of different sequences of genotypes present in the population at the given time, right? This was repeated for many different passages and at many different temperatures. So eventually we get this total, this grand total of values in the region 1, which is in this maturation protein. We got, you see, over 300,000 different sequences, almost 2 million different sequences in region 2, and this figure in region 3. This is, I have to tell you, the same virus. This is summing up all the different sequences we obtained for all the different experiments we have carried out, and for populations that we have sequenced in the symbols shown here. So just a few instances we have. We are now awaiting for further results, but we have sequenced the initial population, passage 4, passage 10, passage 30, passage 60, nothing in between. And the diversity, the genomic diversity that we get for sequences that are not that long, I think it's amazing. So this is different sequences, right, Amir?
Why did you choose these 3 regions? What was the reason?
Well, okay. For some, from the theoretical viewpoint, my expectation is that there are no topological differences between the 3 regions. From the experimental viewpoint, I work in collaboration with people who have been studying this virus. So they are experimental virologists, have been studying these viruses for 2 decades. So they are interested in particular regions. For instance, the RNA-dependent RNA polymerase is a very important protein that is believed to be involved in adaptation to high temperature. They had already detected in the consensus sequences some mutations that seem to be adaptive at high temperature, and this was falling within region R3. Right? This would be the reason for this region here. For this second region, because it overlaps 2 proteins and has an encoding region in between. This is the only section in the virus that has this property. Right? So and this would allow us to compare if there are any differences between non-coding and coding regions, for instance, regarding mutations. And for the first region here, well, this is probably just for comparison, but it was known that there seemed to be an increase in incorporated mutations in this region. And since we wanted to see diversity, we were favoring especially regions that had an slightly over average mutation rate. Right, so this is the reason for these 3 regions. If we would have much more money, we would sequence more regions and yeah, in different proteins, and yeah, that's also a limitation. Yes, Preet?
Yes, can you explain what are the edges in this case? Like we explain it, the edges.
Yes, I was going for it. But yeah, I hope that the explanation on the regions is now clear. I did want to say something else. No, okay. Now let's go to edges. Edges is number of links. And this is, as in the other networks I have reconstructed for RNA. 2 nodes are linked if they are 1 mutation away. Right? So this is the simple reconstruction of the network. So we took all the nodes, and we had a link between 2 nodes if they differed just in 1 position. Right. This is the number of experimental conditions that we have for each of the regions that we have sequenced so far. And this is, yeah, for you to see the sequence length. Here we have a technical limitation. So single molecule sequences cannot go to much longer sequences. So we cannot sequence individual genomes of viruses. They have so many mutations that it's not possible to do shotgun sequencing and reconstruct individual genomes because everything is messed up, right? So in viruses, if we want to see genomic diversity, we have to go to this deep sequencing of single molecules, and then lengths of sequences are necessarily short. Still, the number of possibilities is 4 to the 271 here, which is something that is extremely far away. So a universe time away from the space of possibilities, right? And from the space of what we can sequence in a real population. We can have at most 10 to the 8, 10 to the 9 viruses, and the coverage of our experiments is about 500,000 per experimental condition. So we have 500,000 to 1 million sequences for experimental condition. Of course, many of them are repeated, of course, because they are the same sequence. Okay, so let me now just straight show you how these networks look like. Right, this is a temperature 40 passage 4, And it was not that easy to draw this network because, as you can imagine, nodes were not organized initially in the way I'm showing here, where you can see somehow layers of nodes, right? If you plot them all together, this was a sort of messy galaxy, a planetary nebulous. We will find it fascinating because this is the actual diversity at one point in time in one experimental condition of a viral population. So you have many sequences. Size of the node is the is the abundance of each of the sequences. So many of them are close to this sequence, which is corresponds to the initial condition of the virus. Then you have all this diversity of at distance 1. You see with very different abundances as well. Some of them not that abundant and some of them more. These are distance 2 from the central sequence, distance 3, distance 4, and we go up to distance 5. All right. So since our populations at the beginning were clonal and closely related, they are relatively similar. And in the virus that we sequenced, we didn't find more than 5 mutations difference between sequences in the population. But still, you see that the local exploration is huge because it's occupying many of the adjacent, many of the neighborhood around each of the sequences. And you can also tell that there will be a preference for some regions that are overpopulated compared to other regions where you don't find these sequences, right? We are now studying these networks. We have a large number of results still unpublished, but we believe this is going to be a breakthrough in understanding the diversity of viruses, the real diversity of viruses. So this network to fix ideas is non-neutral because not all genotypes have the same value. Some of them have mutations that change the protein. There are multiple different proteins represented. Again, this is a single population. And the sequence protein are also very diverse. This is out of equilibrium, of course. After 4 passages, we have not reached equilibrium because it's strongly conditioned by the initial condition and has only explored very locally the space of sequences. So this differs very much from the networks that we could study computationally, which were anyway much smaller, but where we had a sort of general picture of the complete topology of the network. And here we are bound to study what happens locally, right? So after 60 passages, things start to change, but you see that the change is slow. Now I don't have one single dominating sequence. I seem to have 2. They are 1 mutation away, that's true, but the neighborhoods are explored also in different ways. So for instance, there is this sequence 1 mutation away from that one, and probably 2 mutations away from this one. So I don't see a link between these 2 nodes. They are not linked because they cannot form a triangle. Typically, there are exceptions. And so I see a broader exploration and I start to see mutations at distance 6 and yeah, I don't know, 6 and 7, right? So this is what I see here. So no, this is just 6. So my population is moving in the space of sequences, but even after 60 passages, it's not that far away from the point where it started. So can you imagine the time it might take to reach mutation selection? So we are still very far from mutation selection. And the question is, does it exist at all? So it exists mathematically, but what happens in reality, if I am bound to explore locally the space of sequences, how long will I need until a new environmental change comes? Will I have time to settle in this equilibrium solution? So my idea is that no. I mean, in molecular evolution, in viruses, we never see mutation selection. We see a sort of optimization of phenotypes, that's possible, but we don't see mutation selection at the genotypic level, which is also against many of the ideas that classically people had regarding the genomic composition of populations, right? So it's not possible to reach that equilibrium because of this network structure and because of the existence of these more connected regions and because of the size, of the very size of the genotype networks. Interestingly, when I look at the empirical degree distribution of these networks, this is what I get. Here I have more data, not just the 2 pictures I have shown you, but 2 more passages at 20 and 30 passages. And I have the joint network with all the empirical data I got, regardless the experimental condition. And you see that this seems to be close to a power law, at least for what we are measuring. We believe that this is the result of the strong hierarchy that is imposed by this local exploration of space, right? I can go from here to my 1 mutation away neighborhood, and from the 1 mutation away neighborhood, I go to the 2 mutations away neighborhood. And this hierarchical structure in networks usually translate into a power law distribution of degree, which is not the power law distribution of the complete neutral network. This is something else, but this can tell us something important about the evolutionary process in actual population whose size is many orders of magnitudes below the size of the space where they could sit, right? Actually, okay. And this is for assortativity, and that's also interesting because the trend in assortativity seemed to be, with the exception of these initial points here, which I believe are bad sampling, insufficient sampling, seems to be disassortative, the network, right? So it seems that nodes of high degree are connected to nodes of low degree. Again, I believe that this can be a local effect where you have a very large periphery with very few links, and these hubs here that link essentially to all other neighbors, right? So you don't have strong connections between nodes in different regions, right? So this would lead to a disassortative network, but this is something that we are still exploring. And also, while the degree distribution didn't seem to change when I was considering independent conditions or the whole network, it seems that in this case, the slope changes when I consider the joint effect of all the sequences I have. This might mean that actually problems of sampling is what make this deviation towards this assortativity. So, but this is something that has to be explored further. And something that we have started to see is indeed how these populations move on the space of sequences. What I'm plotting here is the 100 most abundant sequences in orange, the orange dots with sizes of the dots proportional, of the circles proportional to the abundance of each of the sequences. And the black network below is the joint network of all 100 most abundant sequences in all empirical conditions, right? And this is useful because now when I go through different temperatures and through different passages, I see how populations move in the space of sequences. Again, I have not enough empirical data to see whether sudden transitions occur, but I do see transitions. For instance, here at 43 degrees, you start in this region of my sequence space, and you see how the population changes to another region, which might be better in this environment. And if I go across environments where temperature changes, I see that at 30 degrees, I have a preference for this position here, and I move to another region when I go at 43 degrees, right? So we expect to have soon more data on the dynamics and to be able to quantify better these transitions and see how smooth or how sudden they are, right? Okay. So I still had another history about viroids, and I'll be going a bit fast through it. I don't want to finish without letting you know about these organisms, but yeah, okay, let's get started. So viroids are small RNA molecules that do not code for proteins, but infect plants and cause disease in plants. They just replicate. They replicate and propagate from cell to cell, and somehow also from plant to plant, right? There are many unknowns about these viroids, but it's very interesting that they have also this rod-like structure for its most part, and more complex paw-like structure in another family. There are just 2 families of viroids. Here you see, for instance, the comparison between the T7 DNA and in electronic microscopy, and the size of the potato spindle tuber viroid, which is producing this disease in plants. Right, you see they are extremely small. They have a few from between 250 and 400 nucleotides in length, so very small molecules. It has been argued by they're discovered precisely by Theodor Diener that they might be relics from precellular evolution. So from what we know about these genotype networks and the generation and convergent evolution and so on, I really believe that it's not so difficult to get viroids de novo. So to produce them, to have them emerging in the in a proper, in a suitable cellular environment, and this is what I believe happened in plants at some point, because there were no plants, right, at the RNA world time, and it's I find it really impossible to imagine that a stretch of 200 nucleotides has maintained some memory of its composition and some inheritance of what the original viroid was at the RNA time to our days. I mean, I find it really difficult to believe, so difficult to swallow. So first, you have a structure for free. This is something that we have already discussed. If you calculate how many circular RNA structures of length L with H hairpins and P base pairs are there, this is just the structures, this is what you get. So essentially, many of them have 2 hairpins, 2 terminal hairpins, right? Some of them have 3. This is a branched structure, right? These are circular, so no dangling ends. These are this kind of structures. This would be with 2 hairpins and a more complex one, which have an additional stack and a 3rd hairpin. And so most of them you see have 2 hairpins, right? And so these are the numbers here, the different structures. And although you have with 3 and 4 sometimes, they are not so abundant. So you just randomly fold sequences. You have hairpins, you can catalyze ligation reactions. Therefore, you can have circular RNA with relative easy, right? You can circularize your RNA. And the most things that you get are viroid-like structures, or at least rod-like structures. Right. Also, folded structures are passively selected if they are circular, because they cannot be that easily degraded. So they have an average lifetime, which increases with respect to open structures. And this is one of the things that I think I pointed out yesterday, which is that short stems select a composition that make them resemble double-stranded DNA. And interestingly, in plants, in one of the families of viroids, Pospiviroideae, use DNA-dependent RNA polymerase for replication. So they don't use an RNA-dependent RNA polymerase, but they have just mimicked it so as to use the cellular polymerase, right, for the replication. Right, they are disguised as DNA. Something that we can also do is to evaluate how common or how rare is the structure of viroids, right? So using something that I briefly described, we'll briefly discuss it regarding the degree of neutrality of paired and unpaired positions in RNA folded structures. One can estimate the size of the phenotype as 2 quantities that can be empirically estimated to the power of the number of paired nucleotides and number of unpaired nucleotides. So that's very easy, but this estimation works really nicely. This is the actual calculated size of genotype sizes for length 16 in RNA, and this is the estimated size. And you see that they all fall along the diagonal. So we are pretty confident that this way of estimating sizes of genotype networks for RNA secondary structures is giving very good approximations about the size of longer structures, and in particular of viroids. I mean, viroids have length 200, and it's really impossible to make any good approximation of the size, if not because of this analytical development. Again, a topic that we have discussed a couple of times, which is the existence of a specific sequence motifs which are important for replication. So viroids, some families of viroids have this sequence motif that has to be conserved and is probably involved in replication initiation, right? So what's the likelihood that this motif appears somehow for free? This is something also that we have calculated. So this motif can appear at a given fixed position if you want it to be sitting somewhere specifically in 3 out of 100,000 sequences, which is quite abundant when you have populations that raise 10 to the 8, 10 to the 9. So you have for free, this motif is already present in any reasonably sized population. If the initial position, however, doesn't matter, you find the motif in about 1% of the sequences, right? So I think it's very important when one considers how singular or how selected a structure or a motif or a sequence is, you have to make some calculations to know how far from being just present in a sufficiently large population it can be, right? And this is the case for this motif. So there is no need for a specific selection of the motif along along a long evolutionary time, right? It can be just there for free. So, and then, well, we had some ideas about how to generate, if one would like to do it in the lab, and I hope that someone takes these ideas and does it at some point, to try to generate de novo these viroids. So you just take your sequences and depending on how your initial population is, you either have slight variants or you have random sequences, and you just replicate and select under selection pressures that you think are important for them to start replicating. There are, as you may know, some experiments already where you start with random RNA, and if you put already polymerases inside in your mixture, then sequences start to replicate and you have some evolution towards whatever, right? Okay, so also once you have these small molecules for free, which are rod-like, which can have this motif that you are looking for, that allows it probably to be recognized by a polymerase, then you can trigger selection, natural selection, right? And when this process is on, then you can vary in many different ways, and you will be incorporating advantageous mutations to your molecules. That's for sure. If the niche, the ecological niche, the molecular niche is available. So if there is a role, an ecological molecular role for these molecules to play, right? So you can have increasing length through random addition of nucleotides. You can have recombination. Actually, there are important instances of recombination in small organisms, like in hepatitis delta virus, which has a coding region, but has also a viroid-like coding domain that helps in replication, right? And of course, this is very likely the product of recombination of these two, of modular evolution and recombination of these two differently selected in different environments parts of the molecule of HCV, right? So altogether, we also devised a situation where you would have this random pool of sequences. You could have a circularization of molecules due to the for free presence of hairpins. You can have modular evolution to longer sequences where you could combine coding, even coding segments to non-coding segments to hammerheads and so on. You would have then selection of sequences, some of them for free, some of them just due to the recognition or the interactions with some other molecules in the environment that represent at this point selection pressures, and the joint action of modular evolution plus selective interactions with other molecules. So eventually could lead to a fully developed viroid And my take is that this can happen in relatively short evolutionary time. I would be happy if at some point this is done experimentally, right?
Susanna?
Yep.
Yeah, no, it's all right.
It's all right. Okay.
Yeah, I was actually I'm going to ask you to summarize. And that's
yes, absolutely, absolutely. I didn't want to finish without giving this last example of viroids, but yeah, this is just a summary of some of the ideas that we have been discussing here and how they might be important when we look at how molecular evolution proceeds and what happens at the level of viruses, especially, and RNA and so on, right? So I didn't want to say more, just to let you know that there are many people through the years that have been involved in some of this research, beyond, of course, many other groups that whose results I have been presenting, and I've given you references. But my acknowledgement to Esther, who was doing the experiments with Kavita in her lab. And she's also a long friend of mine for a long time. And also to Jose, same situation, but he's the mathematically talented part of this team. And so he's able to do magic with combinatorics and so on, which is very useful. And all these people have been in my group at some different stages, and some collaborators that also were involved in different, so in helping us understanding what's going on. So, okay, that was it. I don't know if there are questions, or we should stop here.
Yeah, I don't see questions. Okay, so yeah, I don't see any questions. So, yeah, so can we stop sharing the screen? We can just say,
okay,
it's your last lecture today.
Yeah.
Yeah. So thanks so much, Susanna, and it was really nice having you here. I know there were all these family issues and work and so on, but I'm glad that you could make it in spite of all these problems. And
okay,
there were lots of questions. Yeah.
Okay, great.
So,
I hope you enjoyed some parts of it, at least it was profitable for some of you.
Yeah. I think I know some of these things I have heard when I was starting out on quasi species, etc. At that time, I was also reading about neutral networks. So that's how I knew your work already.
Okay. Great.
So, Deepa,
so thank you. Thanks, everyone.
No, it was enjoyable. There. Thank you very much for being here.
Okay, thank you very much.
Thank
okay,
you.
so
bye-bye.
Bye.